Thank you so much for the opportunity to speak here. I'm here, as you mentioned, on behalf of iGEM Leiden for this year's theme. So I'll just dive right into it. For those of you who don't know, iGEM is an international student competition. iGEM stands for the International Genetically Engineered Machine Competition. And it's a competition where teams of students try to tackle a global issue or a local one. Often these are environmental issues, such as dealing with pollution, or it's a medical issue like diagnosing rare diseases, or, well, these complicated issues that could potentially be solved by genetically engineered organisms. So a little bit about me. As you said, my name is Sebastian. I did my Bachelor of Biology here at Universiteit Leiden. After this, I did a Bachelor in Japanese because I wasn't sure what I wanted to pursue further. But eventually I made up my mind, and now I'm in my Master's Molecular Genetics and Biotechnology. Also functioning as the communications manager for iGEM Leiden, which means I'm in charge of managing social media and the email. My personal interests have a lot to do with biology, as you can see, and in my free time, I like to go bouldering and going for drinks with friends and colleagues. So I'll first briefly introduce the background of our research. There's a lot of cool iGEM projects that try to solve complicated issues, such as the nitrogen crisis, oil pollution, bioremediation, sustainable food production, and they all have in common that they do this using genetically or modified organisms. However, there's one problem with this, and that's horizontal gene transfer. Horizontal gene transfer is the transfer of genes between unrelated species. So this often includes bacteria that give their own genes to different bacteria, and the same can happen for your own synthetic genes that you put into your bacteria. So our research project is focused around how we can make the use of genetically modified organisms in an uncontained environment that is outside of a bioreactor, where you can just keep them all safely locked away. How can we make this safe and prevent the spread of these genes into wild organisms and prevent our organisms from moving where we don't want them to be, where they could cause environmental damage or other problems? In terms of how fundamental and applied this is, we are on the fundamental side, as we are trying to make a system that can be the basis for other people to use. And tying into this, the expected impact on society is quite long-term, partly to do with the fact that right now it's very difficult to get a license to use GMOs outside of the lab. So our research is quite fundamental, and we expect it to have a long-term rather than a short-term impact on society. As I'm not sure everyone here has a background in biology, I'll briefly go over a couple of terms that I'll use in my presentation. The first being plasmids. Plasmids are circular pieces of DNA that in the wild are often used by bacteria to pass on antibiotic resistance genes to each other, so that they become resistant to antibiotics that might be in the environment. However, in biotechnology, we use plasmids to genetically alter bacteria, since they can easily be inserted into a cell. They are easy to work with, and yeah, that's why they are used a lot nowadays. Further, we have toxins and antitoxins. As the name implies, these are molecules that can be toxic to the cell, and they often or always come with a counterpart, the antitoxin, that relieves this toxicity. We know that they are important to bacteria because we see them throughout the genes, throughout the chromosomes of a wide variety of bacteria. And researchers are not entirely in consensus yet what their function is, but it's likely that it has to do with stress response and keeping bacteria alive in difficult circumstances. It is believed that these toxins, they can alter the metabolism of bacteria so that when there's a stressor in the environment, like an antibiotic, these bacteria can turn on the toxin and stop cell division, which might be what the antibiotic is targeting. Or it could be response to drought or heat stress, etc. Lastly, we have promoters. Promoters are regions in the DNA that determine how active other genes are. So, for example, a promoter might activate a certain gene or deactivate a certain gene that's right next to it on the DNA. So for our project, as mentioned earlier, we designed a system to keep bacteria in place. We call it DoppelLock because it stands for double plasmid lock. And the idea is that you insert two plasmids into a cell, and both plasmids will make a toxin, and the antitoxin for the toxin will be on the other plasmid. So that if by chance one of these plasmids spreads into a different cell, the cell will only contain the toxin gene and miss the antitoxin gene. This means that it's highly likely that this bacteria will be either killed by the toxin or they will be outcompeted because they carry this metabolic burden that other cells in the environment do not carry. And with this, we hope to ensure that this spreading of plasmids between unrelated bacteria can be drastically reduced in likelihood of happening, which would theoretically make it safer to use them outside of the lab. Next, we also came up with an idea to tie these bacteria to a certain physical space. And we try to do this using conditional promoters. A conditional promoter is a promoter that is only active in specific circumstances. Specific for our system, we use the PBOT promoter, which is only active when there's arabinose in the medium, a specific sugar type, and it is repressed, so it makes the genes inactive if there's glucose in the environment, which is a common sugar. Now, if you put this conditional promoter right next to the antitoxin gene, it will mean that as soon as there is arabinose in the environment, your bacteria can start producing this antitoxin. So as soon as they escape your environment and arabinose is no longer present, they won't make your antitoxins anymore. So using this mechanism, we hope that we can really tie these bacteria into a specific place and also make sure that they don't give their genes, give their plasmids to different bacteria that might be present there. That's a global overview of how it's supposed to work. And now I'll go slightly into detail about what experiments we have been doing over the summer and how we've been trying to make this system reality. Of course, the main thing is trying to actually build these plasmids with all the genes present, selection markers in there, the antitoxins, etc., and get these into the same cell. Unfortunately, over the summer, we have not been able to do this yet. But we do think that it's definitely a possibility. Next, we also had to find a way to balance the amount of plasmids that's present in the cell, because every plasmid has a unique origin of replication, and this origin of replication is basically just a sequence of DNA that determines how many copies of the plasmid is present in the cell. So by having different plasmids in there, you will have different numbers of plasmids present in each cell. This in turn means that you could have a very large difference in the amount of toxin or antitoxin that is made. So we've been designing experiments to approximate the relative concentrations of plasmids in each cell. Lastly, we also had to make sure that the toxicity of these toxin genes are balanced out. So if one of the two would be way more toxic than the other toxin, this could, for example, create a way stronger selection pressure to maintain one of either plasmids and lead to the eventual loss of one plasmid, as well as make it more difficult to balance the copy number of both plasmids. So for those of you who don't know, the general way of making a bacteria GMO is that you make your DNA, then you put your DNA with your cells, and you sort of shock them either with electricity or with heat, and this will make their membranes be able to take up the DNA for a short while. Now, after you do this, these plasmids usually contain an antibiotic resistance gene, and this means that if you then grow those bacteria that you just transformed with the antibiotics present, only those that have your plasmid will survive, and they can grow like this on a nice agar plate. After this, you can then take bacteria from this agar plate and grow them in a liquid culture, and you can extract your DNA again and check if it's correct. Because as it turns out, cloning is a little bit difficult, and it often goes wrong. So here on the right side, you see an example of a gel which contains all these different kinds of bands. And these bands you are seeing, they are bands of DNA. And by checking how long these bands are, you can get an idea of whether your transformation was successful, whether the bacteria you checked are actually the bacteria you're looking for, etc. And here on this gel, unfortunately, these bands do not have the correct length. So our first experimental line did not succeed yet, but we also have other results. The next experimental line was trying to figure out the copy numbers of the plasmids. We did this by inserting a gene for a fluorescent protein into the plasmid, and then growing these cells either with only one plasmid or with a combination of different plasmids, and then evaluating the relative amount of fluorescence to see which fluorescent protein would give the best expression levels relative to the other plasmids. We did this using two different fluorescent proteins, green fluorescent protein and red fluorescent protein. And eventually we found out that two specific origins of replication were the best combination to use, yielding us both a high expression of proteins and an expression that's really comparative to each other. So far we have been able to select and identify which plasmids we would like to use as a backbone for our system. Further, for evaluating the toxicity of our genes, we grew our cells with plasmids that contained our toxin genes in front of the inducible promoter I was speaking of earlier, rather than the antitoxin. So by adding arabinose to these growing cells, we were able to make these cells express these toxic proteins and then check how strongly their growth is impaired. So in these graphs, the bluer the line, the less arabinose is present, and the redder the line, the more arabinose is present. And the growth was monitored over time. On the vertical axis, you see the optical density, which is an approximation for how many cells are present in a liquid culture. To our surprise, however, not all toxins were really as toxic as described in literature. However, for several of the toxins, we were able to detect a significant growth impairment, in particular CCDB, which is a toxin gene that prevents the cell from replicating its own DNA. One of the reasons we think that these cells did not completely stop replicating, but only decrease their growth after a certain time, is that if you grow this in a liquid culture, you can get mutations in your toxin genes, and as soon as one cell line has this mutation and the toxin becomes less efficient, it will grow quicker than the rest and outcompete the other microbes in the culture that do have the toxin still. So this is our current hypothesis as to why these toxins don't completely kill off the cells. However, we have had discussions with some experts on toxins and antitoxins, and they said they weren't completely sure as to why we see this, because it should be lethal to the cells. But that's another interesting result. And from this, we can conclude that there's definitely a strong need to pick the correct toxins, because some of these toxins clearly do not work as well as we would have expected beforehand. Next, I'll tell you something about our future work. We would like to use the system and make it a modular system that can be used by other people, very easily altered to match their exact needs, switch out promoters, toxin genes to meet whatever standards another party might have. Other things we would, of course, like to do is finish actually building the system and testing the functionality before we can make this modular system. But by doing so, we hope to enable other people to take their own synthetic biology projects out of the lab and pave the way for semi-contained synthetic biology applications outside of bioreactors. As Francesca nicely mentioned earlier, we are currently crowdfunding, and we're really looking for some sponsors still. So if you thought this was an amazing project, feel free to chip in, but don't feel obliged, of course. And I would really like to thank all our sponsors who have been really essential in enabling us to do our work in the lab over the summer. And you can sign up for our newsletter. The link is on the left bottom. I will paste it into the chat in a bit. And you can follow us on our social media accounts as well. That was it from my side. And I'd like to invite you to raise your hand if you have any questions, and feel free to respond to anything I said. So thank you very much.